Hi, welcome to my presentation on molecular information flow and protein processing. Most of the information we'll talk about pertains to bacteria. I'll also mention occasionally how things are different for archaea and for eukaryotes. So first we'll go over the basics of nucleic acids and proteins, then we'll get into how they are used to process information. So first we'll do a basic overview of information flow in organisms. Information is stored in organisms in DNA, and their genome is all of the DNA in the cell. So organisms store their information in their genome. Most of the DNA in the genome is in a chromosome, one or more chromosomes. A chromosome is just a big molecule of DNA. Prokaryotes will usually have one, and eukaryotes will have multiple. Within the chromosome, DNA is organized into genes. One gene generally codes for one protein. There's some exceptions to that, especially in prokaryotes. You could have one gene that codes for multiple proteins, and you also have some genes that code for RNA. So in general, the information that's needed to build a protein is found in a gene for that protein, which would be located somewhere on the chromosome in the organism's genome. Then the process of actually using that gene to create the protein that it codes for is called gene expression. So when a cell is actually using its gene to make some product, that is an expressed gene, and that cell is expressing that gene. You have three major processes that cells use to work with the information in their genome. First, replication, which is just the process of copying the chromosome or other piece of DNA. So whenever the cell is going to divide, it's going to make a copy of all of the DNA in its genome, and that process is called replication. When a cell is going to actually express a gene, though, then it goes through the other two processes, transcription and translation. So in translation, the gene is used to make an mRNA copy of the information. The enzyme RNA polymerase is responsible for creating that mRNA copy of the gene. And then that mRNA is going to be used in translation to actually create the protein, and it would be the ribosome that is responsible for that process. The ribosome includes RNA called rRNA. rRNA just stands for ribosomal RNA, and it's actually the rRNA that is doing a lot of the work there of creating the new protein from the mRNA. And you also have other RNAs helping, tRNA in particular. And so we're going to go through these processes in more detail after we go over the basics. But here you have a basic graphic showing how information flows in organisms. So it starts off in DNA, then in transcription, the information is placed into RNA format, then in translation, the information is used to build a protein. So the protein then would have that information. And also you have the process of replication in which DNA is copied prior to cell division. Here's a graphical summary of those three processes of information flow. First, replication. So this would be DNA polymerase that is actually creating new copies of this chromosome. Then you have transcription, in which RNA polymerase creates an mRNA transcript based on a gene. And then finally translation, where a ribosome works with tRNA to read the information on the mRNA and create a new protein. So first we'll go over nucleic acids, then proteins. The two major nucleic acids in cells are DNA and RNA, and the building blocks for both of those are nucleotides. Each has a different set of nucleotides. So DNA has A, T, C, and G. That's adenine, thymine, cytosine, and guanine. And RNA has A, U, C, and G, which is adenine, uracil, cytosine, and guanine. So the main difference would be that in RNA, instead of thymine, you have uracil. Every nucleotide has the same three basic parts. First you have a sugar, then a phosphate group, then a nitrogenous base. In the sugar, the carbons are going to be numbered from 1' to 5'. And there's two types of sugars that you could have. If this is RNA, then we have ribose. If it's DNA, then we have deoxyribose, which is just ribose that's missing a particular oxygen. Actually, here on the second carbon, on the 2' carbon of the ribose sugar, in DNA you have no oxygen, so that makes it deoxyribose. In RNA, you would have an OH group here, so that's ribose. Then the nitrogenous base is where the kind of name of the nucleotide is coming from. So all nucleotides for DNA are going to have the deoxyribose and a phosphate group. All nucleotides for RNA are going to have the ribose and the phosphate group. And then the differences between the different nucleotides would be mostly in terms of the nitrogenous base that's on them. So you have one for adenine, one for thymine, one for cytosine, one for guanine, and one for uracil. So there's 5 different nitrogenous bases that you can have. The base is always attached to the 1' carbon, and the phosphate group is always attached to the 5' carbon. So we say that the 5' end is the front, and then the 3' end is the back. If you're going to have another nucleotide attached to the first nucleotide, it always attaches here at the 3' carbon. And we divide the nucleotides into 2 different groups, purines and pyrimidines. The purines are the nucleotides that have 2 rings on their nitrogenous base. They're adenine and guanine. And the pyrimidines are the nucleotides that have 1 ring on their nitrogenous base, which would be the thymine, cytosine, and uracil. I usually remember it as just purines is the shorter word, but it has more rings. Pyrimidines is the longer word, but it has less rings. Those nucleotides are going to be linked together to form a strand, and the sequence of nucleotides in that strand is the primary structure of the nucleic acid, and that would be where the information is contained in the molecule. So the information is always contained in DNA, the information is always contained in the sequence of nucleotides in a strand, which we also call the sequence of bases in a strand, because the only thing that's different from one nucleotide to another is the base. So here you have a strand of nucleotides linked together. They're always linked with the phosphate group of the next nucleotide bonding to the 3' carbon of the first until they make a chain. And the sequence of the bases is the primary structure where the information is contained in the molecule. The sugars and phosphates form what's called the backbone. There's a special name for the bonds that link nucleotides. It's called the phosphodiester bond, and that would be the bond between the phosphate group on one nucleotide and the 3' carbon on another nucleotide. And we always say that the 5' end of the strand is the front and the 3' end is the back, so we always read it from 5' to 3'. The 5' end would be where you have your free phosphate group that was attached to that 5' carbon. So that's the front. Then at the other end of the strand, you would have an OH group attached to the 3' carbon. If you were going to add another nucleotide to the strand, you would add it down here at this OH group, and that would create a phosphodiester bond. So this is the primary structure for DNA and also for RNA. What's shown in this image here is actually RNA, and you can tell because it has an oxygen on the 2' carbon. If it was DNA, you would just have the H here, no O. DNA has a different secondary structure from RNA. So after the primary structure, that's where the structure diverges for the 2 types of nucleic acids. In DNA, the secondary structure is the double helix, that kind of classical shape that you're used to looking at. In the double helix, you have 2 strands of DNA that are running together, but they're anti-parallel, which means they go in opposite directions. So one of them is running 5' to 3', but the other one would be backwards. It would run 3' to 5'. The bases are going to be sticking into the middle, and they're going to hydrogen bond with each other to form base pairs. So here you have your first strand. It runs from 5' down to 3'. Its bases stick into the middle of the helix, and then your second strand is anti-parallel, which means that at the top you have the 3' end, at the bottom is the 5' end, and then its bases also stick into the middle, and they form hydrogen bonds with the bases from the other strand. So this is called base pairing, when the bases on two adjacent strands are hydrogen bonding with each other, or actually just when two bases are hydrogen bonding with each other. It doesn't necessarily have to be bases on different strands, but in DNA it generally would be bases on different strands, hydrogen bonding with each other to base pair. Then that whole thing is just going to kind of twist up to form the helix. So in order for DNA, two strands of DNA, to form the double helix or the secondary structure, they have to be called complementary, or they have to be what's called complementary, which means that the sequence of the bases on each of those strands has to be such that they would be able to base pair with each other if they were running in an anti-parallel direction. So where you have an A on one strand, you would need to have a T at that location on the other strand, because A pairs with T. Then C pairs with G, so if you have a G on one strand, in that same location you would have to have a C on the next strand in order for it to base pair. So here's an image of the wound up double helix. On the sides here, you can see the backbone, which is the sugars and phosphate groups of the nucleotide strand. In the middle, in yellow and purple, is where you have the bases hydrogen bonding or base pairing with each other. And you have two different grooves on the DNA helix. This one is the major groove, and this one is the minor groove. It's just the space formed between the backbones. When proteins are going to interact with DNA, they will usually bind to one of the grooves, not always, but usually. And they're usually going to be binding to the major groove because it's bigger and there's just more space. A few do bind at the minor groove though, and then some bind at other places as well. So that's the secondary structure of DNA. Then the tertiary structure is DNA packaging. So DNA molecules, chromosomes that is, are very, very large. They're very, very long. They don't fit inside of a cell. For a prokaryote, they don't fit in the cell. For a eukaryote, they don't fit in the nucleus or in the cell. So if you're going to make it fit, you have to package it really well. And different domains of life package their DNA in different ways, but they all have the need to package their DNA. If we're talking about bacteria, then they use supercoiling to pack their DNA, which basically means that you take the double helix, which is already coiled up, and then you continue coiling it until you get extra coils developing, and that's a supercoil. So in this image here, you start out with your regular double helix, two strands coiling around each other, and then you start twisting it and twisting it and twisting it, and eventually you get these additional coils or loops formed by the double helix, and that is supercoils. So here you have a typical image of a bacterial genome. They do package it in different ways, so it doesn't look the same in all different types of bacteria, but this would be one big circle that's been twisted up and coiled up until you get these supercoils, and then it's kind of all held together in the middle there by these special proteins. And the enzyme that is responsible for supercoiling DNA in order to package it is called a topoisomerase. You know it's an enzyme because it ends with the ase. Some topoisomerases are responsible for coiling DNA or supercoiling DNA, and some are responsible for uncoiling it once it's been supercoiled. In archaea, it's a little bit more complicated. First, archaea will wrap their DNA around histone proteins, and then they supercoil it. And in eukaryotes, it's even more complicated again. They're going to wrap their DNA around histone proteins, then coil it, and then supercoil it. And then they may even pack it more if they're trying to divide. So this image here is for eukaryotes. So these are the histone proteins, and you take your double helix and loop it around this core bead of histone proteins. And then in eukaryotes, you're going to form a coil out of those beads and then supercoil that to make this really thick structure. Then if the cell is going to divide, then it's going to package it even more tightly until it forms this condensed chromosome that we're used to seeing. In archaea, it looks a bit different from this. You wouldn't have all of these different histone proteins that the DNA is wrapped around. It would only be like one, and your DNA is wrapped around it and then just super coiled. So you don't have these additional layers of coiling in archaea. So for eukaryotes, you have euchromatin and heterochromatin. Heterochromatin is the more tightly packed chromatin. So if you have supercoiled DNA in eukaryotes, that's actually heterochromatin, very tightly packed, not really active because it's just hard to get up in there and do anything with it when it's that tightly packed. Most of the DNA in the nucleus for a eukaryote would be euchromatin, which is lightly packed. That's just DNA wrapped around the histones and then not additionally coiled on top of that. RNA has a different secondary structure from DNA. In DNA, it's the double helix. RNA can form a lot of different structures for the secondary structure, lots of different types of loops and stuff. In RNA, usually you would just have one strand, not two. So when RNA forms a secondary structure, that means that the strand has kind of looped back around on itself and is base pairing with itself. Which, in order for that to happen, you would have to have different parts of the strand that are complementary to each other in their primary sequence so that they would be able to base pair with each other. So since it's still based on base pairing, it's based on hydrogen bonds between the different bases. So the basis for secondary structure in RNA is the same as for DNA. It's still hydrogen bonding between bases, but just the shapes that you make using that are going to be different between RNA and DNA, mostly because in RNA you're just using one strand that's folded onto itself, and in DNA you have two strands that are coming together. So here's an example of a secondary structure that could be formed by RNA. This is called like a stem loop. So here you have the loop, and here is an area where different parts of the strand were able to base pair with each other, and they have done so, and so that forms the stem. Lots of different little structures similar to this are possible in the RNA secondary structure, and this would generally form just spontaneously by itself when parts of the strand end up near each other that are complementary. Yeah, so that's RNA secondary structure. RNA can also form tertiary structures that are basically more complicated foldings, but we won't get into that. So next we'll talk about genetic elements. A genetic element is just a piece of DNA or RNA. So for DNA, the main genetic element that a cell would have would be its chromosome or chromosomes. Most of the DNA in the cell is going to be inside of one or more chromosomes, depending on if it's a prokaryote or a eukaryote. If we're talking about a prokaryote, it'll probably be just one chromosome that is circular. If we're talking about a eukaryote, it'll probably be multiple chromosomes that are linear. If this is a microbial eukaryote, it probably only has a few chromosomes, but larger multi-celled eukaryotes have many chromosomes. In addition to the chromosome, you also have plasmids possible. Some cells don't have plasmids, some have a lot, it just kind of depends. A plasmid is just a little circle of DNA that's separate from the main chromosome of the cell. Actually, most plasmids are circles, but there are a few that are linear as well, but mostly we think about them as being circles. Plasmids will replicate separate from the main chromosome, and you can have a bunch of different copies of them in a cell. And when a cell divides, it's just going to kind of randomly get different amounts of the plasmids that happen to be in the cell at that time. So when a cell is going to divide, it will specifically replicate its chromosome and give one copy to each of the new cells that's going to form. The plasmids would be replicating by themselves throughout the process of the cell's life, and when it divides, they will randomly end up in one cell or the other. Plasmids end up being important because it can be easy for microbes to share plasmids with each other. When they do that, they're sharing genes with each other, and sometimes those genes might be something very useful for them. A lot of the time, actually, the plasmids can carry antimicrobial resistance genes. They could also carry what's called virulence factors. A virulence factor is a gene that helps that microbe cause disease. So if you have microbes that are sharing virulence factors with each other, then that could mean that microbes that weren't really dangerous for you maybe are becoming dangerous if they pick up a plasmid that has virulence factors on it. Or that could mean that microbes that have resistance are able to share it with other microbes by sharing the plasmid that carries that gene with them. So that's one way that antimicrobial resistance can spread really quickly in microbial populations, especially since plasmids can be shared like between microbes that are of different species. So you don't have, if you're an E. coli and you have a plasmid that has antimicrobial resistance genes on it, you don't have to give it to another E. coli. You could give it to any type of bacteria actually, or archaea also. Then we have finally transposable elements, which are bits of DNA that kind of hop around. So they could be on a chromosome, they could be part of a chromosome, but they can also be kind of cut out of that chromosome and move to a new place. They could move to a plasmid. They could be their own kind of little just chunk of DNA that's floating around waiting for something to like insert into. And they can also be copied, so you can end up with a lot of different copies of them on a chromosome. We consider them separate from the chromosome because they have this habit of like duplicating themselves or hopping around from place to place. There's also a special name for transposable elements, which is transposons. So here you have kind of a summary of different genetic elements in a bacterial cell. So this is the circular chromosome. In this drawing, it's supercoiled, so it's packaged. Then you have some plasmids. Here's one plasmid, here's another plasmid, here's even a third plasmid. So cells can take up plasmids from the environment, they can also release them out of the cell, and then you would just have a variety of plasmids floating around inside the cell. This green chunk here is a transposable element or a transposon. So you can see that's kind of hopping from this plasmid to this part of the chromosome to that part of the chromosome, just kind of hopping around between the different genetic elements of the cell. And in this diagram, it's also showing viruses. A bacteriophage is a type of virus that infects bacteria. So it's showing viruses also inserting their own DNA into the cell. And some viruses would be able to actually like cause their DNA to insert into the chromosome of the cell, so then it kind of becomes like part of the cell, but it could also be like cut out and put into new viruses. So a viral genome can also be a type of genetic, or is also a type of genetic element. In prokaryotes, you have arrangements in the chromosome called operons, which kind of help them organize their chromosome and regulate their gene expression better. An operon is just a cluster of genes that generally would have related functions and also regulatory sequences. And those regulatory sequences are kind of instructions telling the cell like how much to make and when to make it. So helping the cell control how it's expressing those genes. The benefit of having an operon is that you can regulate the whole thing as one unit. So all the different genes that are a part of that operon, you can kind of turn them all on or turn them all off as a single unit without having to have like specific regulatory sequences for everyone and turning them all on at the same time and stuff. So here's a diagram of an operon. This here is the specific operon itself. It has 4 genes on it. These would all be genes that have related functions. Maybe they would be all enzymes for glycolysis or something like that. So when you need one, you kind of need them all. And then in front, we have some regulatory sequences for the operon, and then even some more regulatory sequences down here. These would all be chunks of DNA that are not a gene. They're not going to code for any type of protein, but they are used to regulate how these genes are expressed or how they are turned into protein. And again, you only find that in bacteria and archaea, not in eukaryotes. So that was the nucleic acids. Now we'll go over the proteins briefly. Proteins are also polymers like the nucleic acids, but in this case their building blocks are amino acids instead of nucleotides. Every amino acid has 2 basic parts, the backbone and the side chain. In the backbone you have 3 parts. You have a carboxyl group, a central carbon called the alpha carbon and an amino group. So here you have a basic diagram of an amino acid, of a general amino acid. The R is the side chain. That could be various things. There's about 22 different side chains that are possible for each of the 22 different amino acids. And then the backbone is what's on top. In blue, you have the carboxyl group. Then you have the central alpha carbon. Then on the other side, you have the amino group. The side chains can have different chemical properties. So amino acids are categorized based on the chemical properties of their side chains into 3 or 4 groups. So you could divide them into charged amino acids, polar amino acids, and non-polar amino acids. But then the charged amino acids can also be divided on the basis of whether they have a positive or a negative charge, or whether they are acidic or basic. Since an acidic amino acid would have a negative charge on it generally, and a basic amino acid would have a positive charge on it generally. It depends on the pH, but in an acidic environment with a low pH, that means that you're going to have a lot of protons free. An acid can be defined as a molecule that gives up a proton. Once you've given up a proton, if you're an acidic amino acid, that means you lost a positive charge, which means that you would have a negative charge. If you're basic, on the other hand, a base is something that will gain a proton. So a basic solution would have few protons because they're all being taken up by bases. So if you're a basic amino acid and you gain a proton, that means you gained a positive charge, and you would then have a positive charge. The different amino acids are linked together by special bonds called peptide bonds that are between the carboxyl group of one amino acid and the amino group of the next amino acid. If you have a chain of amino acids that are linked together by those peptide bonds, that's called a polypeptide. Then if that polypeptide is folded up and becomes functional, that's called a protein. But you could also have a protein that has multiple polypeptides in it. If that's the case, then each of those polypeptides would be a subunit of the protein, and the protein is only functional if it has all of them. In order for it to be called a protein, it needs to actually have its function. Oh yeah, and then here in the diagram you have 2 amino acids being joined together by a peptide bond between the carboxyl group of one and the amino group of the other. Okay, so now we'll briefly go through the structure of proteins. They have 4 levels of structure that are possible. They will all have at least the first 3, and some of them have the 4th, some don't. The primary structure of a protein is the sequence of amino acids in the polypeptide. So that's similar to the primary structure for nucleic acids, which is the sequence of bases in a strand. The secondary structure is simple folds of the polypeptide chain. There's 2 different structures that you can make in the secondary structure. You can have an alpha helix or a beta pleated sheet. So in the diagram here, this is the secondary structure. Here you have an alpha helix, and here you have a beta pleated sheet. In each case, these structures are formed by hydrogen bonds between the backbone of different amino acids at different parts of the polypeptide chain. Next, you have the tertiary structure, which is very complicated folding of the polypeptide chain that makes a unique 3D shape that is generally very complex. So this is an example of tertiary structure. Every protein would have its own unique tertiary structure, so it's extremely varied. And there's different interactions between the amino acid side chains that stabilize the tertiary structure. So all proteins will have those 3 levels of structure. When a protein folds, that means that it's going from just having the primary structure to having the secondary and the tertiary structure. And at that point, it would probably be functional, unless, of course, this is something that needs multiple polypeptide chains to act as subunits that all come together in a specific way to make it functional. In that case, that protein would have a quaternary structure. The quaternary structure is how the different subunits are arranged to make the full functional protein. So not every protein has that. Only proteins that have subunits have a quaternary structure. So that's the structure of proteins. Every protein needs to have its correct structure in order to be functional. If it loses the structure, it will lose its functionality most likely. The process of unfolding a protein or having a protein lose its structure is called denaturation. A denatured protein, is one that has lost its structure, which means that it has also lost its function. Specifically, that would mean that it's lost the quaternary, tertiary, and secondary structure. It would still have the primary structure. So here's an example of your folded functional protein that has, you can see it has some alpha helices, it has a beta pleated sheet right here, and the whole thing is folded up to make a complicated tertiary structure. After denaturation, all you have left is this polypeptide chain that has the primary structure. The rest of the structure has been removed, and so has the functionality. So denaturing a protein means unfolding it, and it will lose its functionality. That could be caused by different things. Heat will cause it to happen. Different proteins will have a different tolerance for different temperatures. pH can also cause it to happen, and you can also have specific chemicals that make it happen. Okay, so now we'll go over the process of replication for DNA. Again, this is mostly going to be in bacteria. So DNA replication, generally we think about it as being copying the chromosome, but it could also mean copying plasmids. Plasmids undergo replication as well. When a cell is going to copy its chromosome, that means that it's preparing for cell division. That would be the reason why a cell would do that. Every chromosome has a specific sequence called an origin. At the origin, you have a specific sequence of bases that makes it the origin, and that's where replication will begin. And then you're basically going to split the two strands apart and use each of them as a template to build a new strand off of. So in that case, the original strands are called template strands or parent strands, and the new strands that you're building would be called daughter strands. And you're going to have replication going in both directions away from the origin. So if you start out with just this circular bacterial chromosome, your origin is right here, you would split the DNA helix apart at that point to make a little bubble like this, called the replication bubble. And you're going to start synthesizing a new strand using each of the original strands as a template. And so it would look something like this as you're going away, synthesizing new DNA in both directions away from the origin, and you're just going to kind of progress around the chromosome until you get to the other side, the opposite from the origin, and at that point you have two new chromosomes that you've just made. And this shows it in a little bit more detail. With your origin right here, the two strands will separate, and you start using each of these existing parental strands as a template for a new strand. So you have one new strand being built out this way from the origin. The other new strand would be built out the opposite way from the origin. And it's just going to proceed until you get to the opposite side of the chromosome. That's for bacteria. If you're a eukaryote and you have a linear chromosome, it's a little bit different. You would have multiple origins along the chromosome, and at each one you would start, you would separate the strands and start building new strands based on the old ones. Going again away from the origin in both directions until you meet up with the next little bubble that was associated with the next origin. So the actual process of making those new strands is sort of complicated, or at least there's a lot of different proteins and enzymes that have a role to play. So we had mentioned earlier that DNA polymerase is responsible for building the new strands, but you also have a bunch of other enzymes that have a role to play as well. The first of them is DNA helicase. The job of DNA helicase is to find the origin and separate the two strands from each other, so unwinding the helix at the origin. So first you're going to have helicase come in and bind at the origin and separate those strands. Then you have what's called the replication fork created once the strands are separated. It's just the place where they are separated at, and it forms kind of like a V, so they call it the replication fork. You would have one of those on either side of the origin. and you're actually going to have technically 2 helicases, one on either side of the origin. Then you have single-strand DNA binding proteins, that's abbreviated SSBPs, that come in and bind to the strands where they've just been separated, and they basically just hold them apart so they don't come back and base pair with each other again, just making sure that the replication bubble stays open. You're also going to have another enzyme going in front of the replication fork, unwinding the supercoiled DNA. In bacteria, that's called gyrase, which is a type of topoisomerase. So it's always going to be a topoisomerase, but if it's in bacteria, it's a specific type of topoisomerase that's called gyrase. If you remember either of those, that'll be fine. So either gyrase or topoisomerase, whichever works best for you. So here's your topoisomerase, relieving the supercoiling of the DNA. So, and then this is helicase that's actually opening it. So as replication is proceeding, you're going to be moving further along this original double helix. Helicase is going to be kind of walking along here and separating the strands, and then behind helicase, your SSBPs will come in and bind to this newly separated strand, making sure that it doesn't come back together and base pair with the other strand. Then in front you have gyrase or topoisomerase. What happens as helicase is unwinding the DNA is that you start getting even more coils down the line, which will actually put a lot of stress on the DNA molecule on the double helix. It just gets wound like too tight. If it gets wound too tight, then you could either have potentially the strand could break, but what would more likely happen is that helicase just wouldn't be able to unwind it anymore, and replication would just get stuck, and you wouldn't be able to finish. So that's why you need gyrase or topoisomerase to unwind the supercoiled strand or double helix, unwind the supercoiled double helix in front of helicase. So the way that gyrase or topoisomerase does that is it actually makes a little cut in the backbone of one of those strands in the double helix, lets it kind of twirl around the other one, and then rejoins it after some tension has been released. So after that has happened, you're going to have DNA polymerase come in to actually begin replicating the new strand of DNA, or the daughter strand. There's actually multiple DNA polymerases, and it's specifically DNA polymerase 3 that is going to build the daughter strand. Every DNA polymerase, though, has a couple of limitations. So first, DNA polymerase is not able to begin a strand from scratch. It can only add things to an existing strand. It can't start one itself. Also, it can't add at either end of a strand. It can only add at the 3' end. So that's actually the reason why we say that the 5' end is the front and the 3' end is the back. The 3' end is the only end where DNA polymerase would be able to add new nucleotides to the strand. So it can only go in one direction, and it can only start adding to a strand that already exists, which means that you need something else to actually start the daughter strand, and then DNA polymerase can get going. And what actually is going to happen is you have another enzyme called primase that makes a primer, and then DNA polymerase 3 will add nucleotides to that primer to be the daughter strand. So primase is an enzyme that makes RNA, and it's going to, so this primer that it's going to make is actually a bit of RNA. It's basically just going to be putting in RNA nucleotides that will base pair with the template strand, just a short chunk of these little RNA nucleotides that will base pair with the template strand. So here you have an example of a primer. Here's also another primer. So primase would come in and add these RNA nucleotides for the primer. Then once that is complete, DNA polymerase 3 will come in and find the 3' end of this primer and then start adding nucleotides to it, and that is now, it's now building like your daughter strand. And here's DNA polymerase 3 in the little red oval. You have one more complication for DNA replication, which is that Since it can only go in one direction and the strands are running anti-parallel to each other in the double helix, you end up with a problem, which is that one strand is going the right way and the other strand is going the wrong way, basically. The strand that's going the right way we call the leading strand. The strand that's going the wrong way we call the lagging strand. So the leading strand is the one that would be on top here. That's the one that ends up running from 5' to 3' with the 3' end being in the direction of the helicase. So as helicase walks down the DNA, DNA polymerase is going to always be able to add nucleotides following the helicase. It just builds the strand and follows the helicase. Because the 3' end of the DNA on the leading strand is always pointing towards the helicase. So it can always add there, moving a little bit closer towards the helicase every time. So that's simple, the leading strand. The lagging strand goes the opposite way, and so it's more complicated. In the lagging strand, the 5' end is actually near the helicase, and the 3' end is away from the helicase. And you can only add to the 3' end. So that means that DNA polymerase 3 would need to build the strand backwards, moving away from the helicase instead of following the helicase. So when it starts off, the helicase will have only opened a little bit of the DNA, and the polymerase, well, primase would first come in and make a primer, then DNA polymerase 3 will come in and start adding nucleotides to that new strand. But that new strand, instead of going to be moving like we're being built towards the helicase, it's actually being built towards the origin, which means that you can only build a little bit and then you run out of space. So you end up synthesizing the lagging strand in fragments, which we'll talk about more right now. First, we'll go over the situation for the leading strand in a little more detail, because that's more simpler. So for the leading strand, if you imagine your origin would be a little bit back from here, you would have had helicase come in, open it up, single-strand DNA binding proteins would come in and bind that. You can see them right here, holding the two strands apart. You would also have a gyrase or topoisomerase in front of helicase, relieving tension on the double helix. And then you're going to have your primase come in to this beginning part of your newly opened helix, like right by the origin. And that primase will make a primer out of RNA, and then DNA polymerase 3 can come in and start adding nucleotides to the primer, and it'll just add to the 3' end following the helicase. Since it's the leading strand, the 3' end always points towards the helicase, and DNA polymerase 3 can always just add in the direction that the helicase is going, and they'll both kind of proceed together all along the chromosome until they get to the end. The way that this is actually happening is that DNA polymerase will just kind of wait for nucleotides to come in. Nucleotides that are just kind of freely floating around in the cytosol will kind of come in here to these free strand, and they'll base pair with the bases on that strand. If the base pairing is correct, then DNA polymerase will catalyze the formation of a phosphodiester bond between this new nucleotide and the existing strand, which is shown down here. Each of these free nucleotides actually has 3 phosphate groups on it, not just one. So when you're adding a free nucleotide to the existing nucleotide strand, then you need to cut off those last 2 phosphate groups. And those are pretty high energy bonds, so when you break them, you have energy being released. And that is actually the source of energy for this process. So DNA polymerase 3 doesn't need to use like ATP to get its energy. It's just going to get energy from breaking off the last 2 phosphates of each free nucleotide. Then it's going to add that remaining phosphate to the 3' carbon of your existing strand, and that gives you 1 more nucleotide added to the strand by a phosphodiester bond. So the nucleotide is going to kind of float in, just base pair. If the base pairing is right, then DNA polymerase 3 will add it to the new strand by making a phosphodiester bond. And it just follows the helicase until it gets to the end. And then it's almost done, but you still have one problem, which is this primer that's made out of RNA, not DNA. And you can't have like little chunks of your new strand being made out of RNA instead of DNA. It needs to all be DNA. So you need to get rid of this primer now. And you have an enzyme called DNA polymerase 1 that is going to come in and do that. It just removes these RNA nucleotides and replaces them with DNA. Once that is done, you have another enzyme, DNA ligase, that's going to make a phosphodiester bond between the newly replaced primer and the rest of the daughter strand. So DNA polymerases can't join two strands together. They can add new nucleotides to an existing strand at the 3' end. But if you have two strands already, you can't put them together using a DNA polymerase. So once your DNA polymerase 1 has removed this RNA primer, it's going to start adding nucleotides, and it'll just replace each of the nucleotides until it gets to the beginning of the daughter strand, and then it's not able to add this newly synthesized bit of DNA to the existing daughter strand. And that's why you need DNA ligase to come in and just make a phosphodiester bond right there. And now you're done and you have a complete daughter strand. So that's the leading strand. Then the lagging strand is more complicated, as we mentioned, and we said it's synthesized in little chunks. The name for those chunks is Okazaki fragments, after the guy who discovered them. Each of those fragments is going to have its own primer. So you're going to have primase come in and build the primer for each of those Okazaki fragments. Starting with the first one, then DNA polymerase 3 will start adding nucleotides to the end of that fragment, and it'll just go as far as it's able to go until it hits the next fragment, until it hits the primer of the next fragment, actually. Then you're going to have DNA polymerase 1 come in and replace every primer with DNA, and then DNA ligase needs to come in and join every newly replaced primer to each other Okazaki fragment, basically. So if you're starting here, this is one of your, here you have one Okazaki fragment, here you have another being built. The primer for this one would be further upstream. You can imagine this just being kind of zoomed in from here. So this would be maybe your first Okazaki fragment. This would be your second one. You would have primase make this primer, which is also this primer. Then DNA polymerase 3 will come in and start adding nucleotides until you get to, well, basically until you get to the origin. At that point, you will run into the strand that's being synthesized on the other side of the origin, and you'll have to stop. Meanwhile, primase will have hopped down. DNA helicase will have opened up a bit more of the double helix. Primase will hop down to the newly opened region and make another primer. DNA polymerase 3 will come in and start adding nucleotides to the end of that primer, making another Okazaki fragment, and it'll go until it hits the first primer, just proceeding until it runs into the first primer. And here it's run into that first primer, so it's kind of done now. That's as far as it can go. Meanwhile, the helicase has moved even a little further along the double helix, and you have another region opened up, so primase will make another primer, then DNA polymerase 3 would come in and start building another Okazaki fragment until it hits the primer from the second fragment, so on and so forth. Then you're going to have your DNA polymerase 1 come in and remove each one of these primers, replacing it with DNA. So it would just be cutting out these RNA nucleotides and then adding DNA nucleotides to the end here of this strand. And this strand is actually from the second Okazaki fragment until it runs into the first Okazaki fragment. And at that point, it's not able to join those two existing strands together. So then you have DNA ligase come in and actually do that, making a phosphodiester bond between the second and the first Okazaki fragments until you have just a smooth region of the daughter strand. And that process is going to repeat all along the entire lagging strand until you have a complete daughter strand. So we've kind of been talking like all these proteins are separate from each other, but the truth is most of them are not. They are kind of all bound together in a giant complex called the Replisome. In order to do that, you kind of have to loop the lagging strand around. So you would have topoisomerase here unwinding the DNA ahead of your helicase right here, which is opening the double helix. Then attached to this helicase, you're going to have primase making primers, and you're going to have DNA polymerase 3, one for the leading strand and one for the lagging strand, and your SSBPs. In order for DNA polymerase 3 on each strand to be like next to each other and part of the same protein complex, you're going to have to loop one of the strands, and that'll be the lagging strand that loops. So it loops around so that DNA polymerase 3 can always be attached to the other DNA polymerase 3 on the leading strand. You do not need to know the details of the replisome, actually. So that's the basics of DNA replication. In general, the DNA polymerase 3 will only add a nucleotide to this daughter strand that it's building if the base pairing is correct between that new nucleotide and the template strand. But sometimes it makes a mistake and it puts a base pair, or it puts a new nucleotide that doesn't base pair correctly. When that happens, it's able to actually check itself and check that it made a mistake and fix it. And that's called proofreading. So DNA polymerase 3 doesn't just build a new strand, it proofreads the strand that it's building, making sure that it doesn't make mistakes, and if it finds a mistake, it will fix it. It can find mistakes because when you have a base pair that isn't right, the shape of it will be off. It'll be kind of bulged out a little bit because two bases that aren't supposed to hydrogen bond with each other or that aren't supposed to base pair with each other, they're not going to be able to base pair in a way that is shaped correctly. So DNA polymerase 3, if it senses that there's the wrong shape of that base pair, it's going to cut out that wrong nucleotide and just put the right one. It'll actually wait for the right one to kind of float in and base pair. Then it'll see the base pairing is right now, the shape is right, and it'll add that new nucleotide to the strand. So here you have a diagram of that. This would be a mismatch. A base paired with C, that is not correct. A should be going with T, not C, and the shape of this is going to be wrong. So DNA polymerase 3 will kind of sense that the shape is off here. It cuts off the C. It cuts the phosphodiester bond between the C and the A that it had already previously added. That C will leave, and it's just going to kind of wait until a T floats in, base pairs correctly. Then it's going to catalyze phosphodiester bond formation between this T and the A. So it's able to fix like most of the mistakes that it makes in that way. Actually, there are still a few mistakes that get through, or that can get through. The cell has a lot of mechanisms for fixing mistakes in its DNA. So even if DNA polymerase 3 misses some of its mistakes, there's still other mechanisms that will catch most of them. But even after that, there's still a few mistakes that don't get caught usually. And each of those mistakes that doesn't get caught, that makes it through every proofreading or DNA fixing mechanism that the cell has, all those mistakes become mutations. Right? That is something that is going to be copied out next time you do DNA replication. And it's going to be ending up in your mRNA for whatever gene it's a part of. And that's basically a mutation at that point that's going to be passed down to all the daughter cells of this cell. So for each organism, it's usually not good to have mutations. Most mutations aren't beneficial. Some of them are neutral, but most of them are bad for the cell, actually. That's why the cell has so many mechanisms for fixing these mistakes and making sure that they don't turn into mutations. But of course, the population is benefited when you do have some mutations, because if you don't have mutations, then you can't have evolution, you can't have adaptation to the changing environment. Just a point. Okay, so that's replication. Now we'll go through transcription. Again, mostly in bacteria. So transcription is the process of making RNA from DNA. Usually we think about transcription being where you're making mRNA based on a gene, but technically anytime that you're making RNA from a gene or from DNA, that is transcription. So, right, genes that code for rRNA or tRNA, those are also going to be transcribed. But most genes code for protein, and you would make first an mRNA transcript of that gene and then use the mRNA in translation to build a protein. But the process of transcription is the same, no matter what type of RNA you're making. It's always going to start at the promoter. Every gene or every operon will have a promoter in front of it, which is a specific sequence of nucleotides that signals that this is where transcription needs to start. So RNA polymerase, which we mentioned is the enzyme that does transcription, RNA polymerase is going to bind to the DNA at the promoter, and then it'll begin transcribing the gene. How RNA polymerase binds the promoter is different for eukaryotes and bacteria. In bacteria, RNA polymerase has a subunit called sigma factor that will bind the promoter. And that's shown in this diagram here. So here you have RNA polymerase. In the pale, I guess really pale purple, you have the sigma subunit of RNA polymerase. And the sigma subunit is what's actually binding the promoter here. And this is the promoter. This would be the sequence of the promoter for different genes. So different genes will have a different nucleotide sequence in their promoter. But you're generally going to have a couple of regions that are the same or almost the same between different promoters. And those are the regions where sigma is actually binding. And you actually don't have just one sigma factor. You're going to have several different versions of the sigma factor. Each RNA polymerase could have any of those several versions of sigma factor as part of it. Since all promoters are similar, but they are also like a little bit different from each other, some sigma factors will bind really well to some promoters, but less well to others. So that's a way that the cell can control its transcription. If it wants to make certain genes, it'll make a sigma factor that binds really well to the promoter for those genes, and that means you're going to transcribe more of those genes. If you don't want to make those genes, then you're going to start making a sigma factor that doesn't bind well to those promoters, or you're going to stop making the sigma factor that does bind well to those promoters. That way it's hard for RNA polymerase to even attach to the promoter in the first place, and if RNA polymerase can't attach, then it can't make the mRNA. In replication, each strand in the double helix, each DNA strand in the double helix is being used as a template strand to build a new strand, but in transcription, you're actually only using one of the strands. And for different genes, it could be one or the other. It kind of switches. It's not like always the left one or always the right one. It depends like on the individual gene. So the RNA polymerase always has a way to tell like which strand it's supposed to be using as the template. The other strand that is not used as the template strand is called the coding strand. It could also be called the complementary strand, just depending on the source, I guess. The reason why it would be called the complementary strand is because it's complementary to the template strand. The reason why it would be called the coding strand is because it's containing a nucleotide sequence that is the same as the nucleotide sequence of the RNA that's being built from the template strand. And that's just because both that coding strand and the RNA that you end up making are going to be complementary to the template strand, right? They're both able to base pair with this template strand. The only difference between the coding strand sequence and the sequence of the RNA that you make is that since it's a DNA strand, you have T, and the RNA that you make, you have U instead. So here's an example of that. Here's your DNA that you're starting out with. These are the two strands for this gene. You're going to make one of them. One of them will be the template strand, the other the coding strand. So in this case, this bottom strand is being used as the template, and the top strand is used as the coding strand. So RNA polymerase is going to come in and start building RNA using the template strand as a guide for which nucleotide to put. Then if you look at the sequence for this RNA strand that it's building and the coding strand, you see that they are the same, except you have U's instead of T's. So in the coding strand, you have ATG. In the template, in the RNA that you make, you have a UG, just a T, a U instead of a T, and otherwise it's the same. So if you read the coding strand sequence, you will know the sequence of the RNA that would be built from that gene. You just have to make sure that every time there's a T, you put a U instead. So once you have RNA polymerase binding the promoter using sigma factor, transcription can actually begin. First, RNA polymerase will open the double helix. So DNA polymerase can't open the double helix. It has to have a special enzyme helicase to do that. But RNA polymerase is able to do it by itself. So it opens the double helix, and that creates something called the transcription bubble, which is just the little region where the helix is open, which is right here. The transcription bubble is always going to be like about as big as RNA polymerase. As RNA polymerase moves along the gene, it's going to continue opening the helix, and as new areas of the DNA come out the back of RNA polymerase, they're going to re-base pair with each other and reform the double helix. Then RNA polymerase is going to start building an RNA strand based on the template strand. So RNA polymerases don't need to start with another strand. They can make a strand from scratch. So you don't need a primer here. RNA polymerase will just start building the RNA strand. And again, the way that it does that is it kind of just waits for nucleotides that are floating around to come in and base pair with the template strand. And once it senses that a nucleotide has base paired correctly, it's going to catalyze a phosphodiester bond formation between that new nucleotide and the rest of the RNA that it's building. And it'll make sure that it's only adding RNA nucleotides, not DNA nucleotides. Again, those RNA nucleotides, when they're free, are going to have 3 phosphates on them. They have 3 phosphates when they're free. So you have to cut out the last 2. And that's the source of the energy for this process. So RNA polymerase is getting its energy from the same place that DNA polymerase is getting its energy from, just from cleaving off the last 2 phosphates of each free nucleotide that is adding to a strand. Also similar to DNA polymerase, RNA polymerase can only add to the 3' end of a strand. So it starts building a strand from scratch, but it can't add to the 5' end of the strand that it's building. It only adds to the 3' end. And RNA polymerase is going to continue walking along the gene, adding new nucleotides to the RNA that it's building as it goes, until it gets to the end. And it actually might end up going a bit past the gene or a bit past the operon. It's actually going to go until it gets to a specific sequence of nucleotides called the termination sequence. Once it hits that sequence and transcribes it, transcription will stop. There's a lot of different mechanisms for how exactly transcription will stop, so we're not going to talk about the details of those. Just there's multiple ways for it to occur, but once the termination sequence has been transcribed, something will happen that causes RNA polymerase to let go of the DNA, and then transcription is done, and you have RNA polymerase leaving the helix comes back together, so the transcription bubble rebase pairs, and then you have this separate strand of RNA that you just made. So this is your RNA product, which is probably mRNA. Okay, so that's DNA. We'll talk about how transcription is different for archaea and for eukaryotes now. It's mostly like DNA, but there are a few differences. The main difference, the biggest difference by far, is how transcription is initiated and how RNA polymerase binds to the promoter. So in DNA, you have sigma factor. Sigma factor is a subunit of the RNA polymerase, and it binds to the promoter, and that's how the RNA polymerase attaches. But in archaea and in eukaryotes, you do not have a sigma factor. Instead, you have other proteins that are not actually part of the RNA polymerase, so they are not subunits of the polymerase. They're independent proteins, and they're called transcription factors. So those transcription factors will bind to the promoter, then RNA polymerase will bind to the transcription factors, and then it'll begin transcription in basically the same way that it works in bacteria. In archaea, there's just a few different transcription factors, but in eukaryotes, there's really very, very, very many. So here's a promoter for archaea, and it has like several sequences within it that are common to find in different promoters. Every promoter is going to be a bit different for every gene or every operon, but you're going to have several kind of motifs that show up again and again in different ones, although not literally in all of them. And it's these motifs, these specific sequences where the transcription factors would be binding. So here you have the TBP transcription factor and the TFB transcription factor. You don't need to know the names of them specifically, but a couple of different transcription factors that have bound to the promoter at these special regions. And then RNA polymerase is going to find those transcription factors and bind to them. In eukaryotes, you have way more transcription factors. Really, sometimes it feels like almost a limitless amount. It's not actually limitless. It's just extremely large, and you can have like multiple that are needed for transcription for any given gene. So here's a eukaryotic promoter. It'll have some characteristic motifs also, some sequences that you find in a lot of different promoters, different ones for different transcription factors. And you'll have a variety of transcription factors that come in and bind to different parts of the promoter, and then RNA polymerase will bind to them. But in this case, here you have 3 transcription factors, and that's not even actually enough. You still have to have this one come in and bind before RNA polymerase can actually attach. So you might have like a number that have to all be bound at the promoter at the same time for RNA polymerase to be able to attach to them and attach to the promoter and begin transcription. You all probably don't know it, but you all know examples of transcription factors because you know of estrogen and testosterone. Those are both transcription factors. So the way that they cause men to develop differently from women is by going into the cells and binding as transcription factors to different promoters, so that a man's cell will make different genes into proteins than a woman's cell would. And that creates the most of the sex differences. Finally, after you have made that mRNA transcript, you're not quite done yet. You have to do RNA processing to process the transcript so that it's mature mRNA. Actually, RNA processing is going to occur in bacteria, archaea, and eukarya, but we're mostly going to talk about it in eukarya because it's a lot more complicated for eukaryotes. It's just much simpler for bacteria and for archaea. So in eukaryotes, you have something called RNA splicing, which makes the process kind of complex. RNA splicing means that once you've made your mRNA transcript, there's sequences in it, like chunks of it, that need to be cut out, and then the ends need to be joined back together, from where you had cut. So a lot of eukaryotic genes need to be spliced like this. Some don't, a lot of them do. And actually in archaea, you do have a few genes that need to be spliced, but most of them don't. And in bacteria, you don't have any splicing. So we'll divide the sequences of the mRNA into exons and introns. Exons are the part that you're going to keep, and introns are the part that you're going to cut out. So every gene can have a different pattern of exons and introns that end up in the mRNA transcript. So here you have an example of an mRNA transcript in a eukaryote that was just now made through transcription. And in red, you have the intron regions, and in, I guess, like orange, yellow, you have the exon regions. So these exons you're going to keep, the introns you're going to get rid of. I always remember it as like the opposite of what you would think. Exon sounds like exit, so it sounds like that's what's going to be leaving, but no, that's what's staying. So exon is for staying for some reason. I'm not sure why they named it like that. So you're going to cut out these introns and join the exons together. So here we've had a couple of these introns cut out, and then the ends of the exons would need to be joined back together, right? So you cut this intron out and this intron out, and then exon 1 needs to be attached to exon 2 needs to be attached to exon 3, and there you have your final transcript. So there's actually different ways to do that. You're not necessarily going to cut out every single intron, which is what makes it like really complicated. Sometimes you might cut out introns 1 and 3. Another time it might be introns 2 and 4. In each case, you're going to be ending up with a different mature transcript, and then that's going to be translated into a different protein. So in eukaryotes, the same gene can be used to make multiple different proteins depending on how it's spliced, which introns you're cutting out, and which ones you're leaving behind. Yeah, so it can get really complicated. In addition to the splicing, in eukaryotes you have a cap and a tail that need to be added to every mRNA transcript. On the front end of the transcript, the 5' end, you have a 5' cap that will be added. The 5' cap is a guanine, but it's like a modified guanine, and you're also adding it backwards. So usually you would add, 3' to 5', but in this case you're joining the 5' with the 5', which you don't need to know, actually, but just 5' cap is fine. The point of adding this cap is so that the ribosome can recognize the transcript later on for translation. Then at the other end of the transcript, at the 3' end, you have the poly A tail that's going to be added, and it's just a bunch of A's, a bunch of adenines that will be added to the 3' end. The purpose of the poly A tail is to protect the transcript from being degraded. So eukaryotes, I guess I will say that they're aware. Of course, they're not really aware because we're talking about just cells, but eukaryotes are aware that viruses may be infecting them. If viruses infect them, then you're going to have viral RNA inside the cell, and that RNA is going to be trying to use a ribosome so that it can be made into a protein. Because viruses can't make their own proteins, they have to have the host cell make the protein for them. In order to do that, they have to use the machinery that the host cell uses, which is the ribosome, so they need to have RNA that the ribosome can use. So eukaryotic cells know that this may be happening, and so they degrade RNA if they find it. So if a eukaryote finds just a bit of RNA floating around, it's going to figure that came from a virus and it's going to destroy it. But it won't destroy it if it sees the poly A tail. If it sees the poly A tail on that bit of RNA, then it's going to know that's my RNA, I made that, and it won't degrade it. So having the poly A tail protects mRNA transcripts from being degraded before they can be turned into protein. So after you've added the 5' cap and the poly A tail, and you've spliced the RNA by taking out the introns, then you have final mature mRNA that's actually ready for translation. And the last thing we'll talk about is translation. So again, this is going to be mostly for bacteria, with a few differences for archaea and eukarya, but archaea and eukaryotes would do it mostly the same way that bacteria do it. They're all going to be using tRNA for this process. tRNA is responsible for kind of actually reading the mRNA and making sure that the right amino acid is brought in and put in the right location for this growing polypeptide. So in mRNA, the information is in the primary structure, the sequence of bases in the mRNA, and you're going to chunk that up into codons. Every 3 bases is 1 codon. So a codon is sort of like a word in the genetic code, and each codon is going to have a specific tRNA that will bind with it, and that tRNA will have a specific amino acid on its other end. So the tRNA is responsible for linking the mRNA information to the actual amino acids. Every tRNA has this strange structure. This is what the tRNA actually looks like when it's all folded up with its secondary structure. But a lot of the time in diagrams we draw it not all folded up because that's like too complicated. So you'll see it drawn as this kind of cross or clover shape a lot. At one end, it has what's called an anticodon, which is 3 bases that are supposed to base pair with the codon. At the other end, it's going to have the amino acid acceptor, and an amino acid would be attached there. So at one end, you have the anticodon that base pairs with the codon. At the other end, you have the amino acid. Every tRNA is going to have a specific anticodon. Every tRNA that has that particular anticodon will have the same amino acid attached at its other end. So that way, when this tRNA base pairs with this specific codon, you'll be bringing in this specific amino acid. So that's kind of the way the code works when you're translating from nucleic acid codons to protein amino acids. And you have special enzymes that are designed to attach that amino acid to the proper tRNA. Those are called aminoacyl tRNA synthetases. You have one for every tRNA. So this aminoacyl tRNA synthetase is going to recognize a specific tRNA that has a specific anticodon, and every time it binds to that specific tRNA, it's going to bring in a specific amino acid and attach it to the acceptor end. So in this case, this tRNA has AAG as its anticodon. Every time you have one specific aminoacyl tRNA synthetase that is going to be binding to only tRNA that has the AAG. Once it binds to that tRNA, it's only going to bring in the phenylalanine amino acid. It won't bring in anything else. That process requires ATP, and it's called charging. So once the tRNA has its amino acid attached to it, we say that it is charged. So the tRNA is responsible for kind of reading the code of the mRNA and then bringing in the appropriate amino acid for every codon. So the genetic code is just all of the different codons that you can have in mRNA. Each one will go with a specific amino acid, with a couple of exceptions. So most of the codons are coding for an amino acid, but you also have a start codon. The start codon does code for an amino acid as well. It codes for methionine, but it also codes for start. So when the ribosome is kind of starting this whole process up, it's going to be looking for the start codon, and it'll begin making the protein from that point. Then at the end, once it's gone through the whole mRNA, it'll be looking for a stop codon that will tell it to stop. So you have 3 different stop codons. They do not code for an amino acid, they just mean stop. Whenever the ribosome comes to a stop codon, it's just going to stop translating the protein. And at that point, the protein is done, or I should say the polypeptide is done. It still needs to fold to be a functional protein. So you would have a tRNA for every one of these codons except for the stop codons. Every codon that has an amino acid associated with it will have a tRNA that has an anticodon that can bind that codon. We say that the genetic code is degenerate. That means that you have, for any given amino acid, multiple different codons that will mean that amino acid. So every codon has only one amino acid that it means, but every amino acid will have multiple codons that it means, that specify it, with the exception of methionine, which is only for the start codon. So in this table here that has the genetic code, you can see that here the codon UUU codes for the amino acid phenylalanine, but the codon UUC also codes for phenylalanine. So in order to make a phenylalanine, you would need either one of these codons. That would be fine. And every amino acid here is going to be here multiple times, generally like even 4 times, 2 to 4 times in general, with the exception being methionine, which is start and methionine. The reason why you have that feature of the genetic code is actually because of the way that tRNA anticodons base pair with the codons. So here you have a tRNA approaching the mRNA. here's a codon in the mRNA, and here's the anticodon of the tRNA. So the first letter in the codon is U, and the anticodon has an A to base pair with it. Then you have C, so the anticodon has a G. And then you have, let's say, a C as the last one. But it actually says C/U here because either one, either a C or a U, would be able to base pair with the anticodon in this case, which is G. So G should pair with C. It shouldn't pair with U. But what happens here is that these little three bases are kind of at the end of this loop, and the third one is kind of actually curving away. It's actually diagonal. It's curving up to the side of this loop. So because of that, it can't properly base pair anyways. So this G would never be able to make a proper base pair with the C in the codon because it's at an angle. It's not hitting it straight on. That means that it would also be able to get away with base pairing with a U too. So that is wobble pairing, where you have this last base in the anticodon at an angle that prevents it from base pairing properly, and also allows it to base pair with multiple bases in the codon. That is wobble pairing. And that's the basis for the degeneracy of the genetic code. So if you have a C here, UCC, that's going to pair with this anticodon specifically and bring in a specific amino acid. If we look it up, UCC codes for serine. So this anticodon would have a serine, well, this tRNA with this anticodon AGG would have a serine attached to it on the other end. On the other hand, if you had a U here for a UCU codon, this anticodon would still base pair with that and bring in the same serine. So UCU, you can see also means serine. UCA means serine as well. UCG means serine as well. So whatever base you have here in the last position for the codon, it will be able to base pair with this last G in the anticodon because that G is crooked. And anytime that happens, a serine will be brought in. This is actually a good thing for the organism as a whole, because it means that if you had a mutation there to that last base in the codon, right? If you started with a C, but then you got a mutation and now you have a U there, that's okay. It doesn't actually affect the protein that you're building, because the same tRNA is going to base pair with that codon either way, and it's always going to bring in the same serine. So mutations that occur to the last base in a codon usually are what we call silent. They don't affect the structure of the protein, and they don't affect the organism in any way. In order for a mutation to affect things, it would probably need to be in the first base of the codon or the second base of the codon. If you change one of those, then you're going to change what tRNA anticodon binds there. That means you're changing what amino acid is going to be part of that protein. Okay, so now we can start talking about the actual process of translation. The first part of translation is translation initiation, and most of this information here is applying to bacteria. And I kind of have notes where eukaryotes or archaea have a different setup. So the first thing that needs to happen in translation is the ribosome needs to get put together on the mRNA. And that is basically what's occurring in translation initiation. Everything's just assembling. And this process does cost energy. It costs GTP, which I think mentioned before in the metabolism chapter, that the Krebs cycle actually technically makes GTP, not ATP. They're really similar, and it basically works in the same way. You just cleave off the last phosphate, and this would be guanosine triphosphate, and that releases energy. So it functions the same as ATP. This process of the ribosome assembling onto the mRNA is like a little bit more complicated than what you might expect. First, every ribosome has two major subunits. Actually, every ribosome is extremely complicated, a giant protein complex with many, many subunits in it, and with different pieces of rRNA in it as well. But we kind of divide it into two big subunits, and we call one of them the large subunit and the other one the small subunit. That just means it's small compared to the large one. It doesn't mean it's actually small. It itself is a very large protein complex with many different proteins and multiple rRNAs inside of it. But in general, we just think about a ribosome as being the large subunit and the small subunit, even though they're both real big. So the first thing that's going to happen is the small subunit attaches to the mRNA. Then you're going to have a special tRNA come in and base pair with the start codon. So in bacteria, that tRNA has a special methionine on it, which is modified. It's formal methionine, so it's methionine but with a formal group on it. Any other time in the mRNA, if you have that start codon again, it's just going to put a regular methionine, not the formal methionine. Only the very first start codon where you actually start, that's the one that gets the formal methionine tRNA in bacteria. In eukarya and archaea, actually, you would be using just regular methionine for that start position. Then you also have some other proteins that have to come in that are called initiation factors. So those have to bind to the small subunit. Those are not shown in this diagram here. This diagram has mRNA, it has a small subunit attaching to it, and then it has the special tRNA, the initiating tRNA, with either methionine or a formal methionine, depending on if this is bacteria or something else. You would also have initiation factor proteins at different parts, binding to different parts of the small subunit, but they're not shown. You need several of them. Those are not actually considered a part of the ribosome, since as soon as initiation is over, they're just going to let go. Then after all of that has happened, you spin some GTP and the large subunit will attach to this whole complex. So now you have basically the whole ribosome completely assembled onto the mRNA with your starting tRNA inside of it in the correct position. The cell has to be really careful that the ribosome assembles on the mRNA in the correct location. It has to assemble so that the initiating tRNA can match up perfectly with the start codon. If you assemble it in the wrong spot, you're going to get the wrong protein. So it's really important that that happens in the right location. In order to assure that it happens in the right location, bacteria have a sequence in the mRNA called the ribosome binding site, or the RBS. It's indicated right here in this diagram. So you would have a sequence of nucleotides in the mRNA here that are the RBS. And so the small subunit is going to be looking for that RBS to bind there. Another name for the RBS is the Shine-Dalgarno sequence. So a lot of the time it's called Shine-Dalgarno after the people who discovered it. So the small subunit will always attach to the RBS, and once it has attached to the RBS, it's always going to end up positioned so that the start codon is like perfectly in the middle where it needs to be. You actually have some rRNA in the small subunit that is going to base pair with the RBS. So that's how the small subunit is attaching to the RBS. It's actually by base pairing with one of the rRNAs that's in there. And this is actually for bacteria. It's also for archaea, but eukaryotes have a different process. They would be using the 5' cap instead. So the ribosome will attach to the 5' cap, and whenever it does that, it ends up lined up in such a way that the start codon is correctly positioned inside the small subunit. And then this diagram here is just giving you an example of what would happen if the ribosome attached at the wrong spot and you didn't have the start codon lined up right. If that happens, you're going to end up reading all of the codons wrong. So let's say that you were lined up so that the initial tRNA didn't bind with the AUG, instead it bound with UGG. First off, you would have to have an entirely different tRNA coming in to base pair that way with that other codon. But that means that every other codon is going to be read wrong then. It's all going to be shifted one base off so that it's all wrong. If you're reading it correctly, you would chunk the codons like this maybe. ACA, UAC, CGA, UCA. If you do that, you're going to get these proteins. this threonine, tyrosine, arginine, and serine. If you had attached to the mRNA wrong, so that you were actually at the UGG instead of AUG, then you would get this, you would chunk the codons like this. So you would have, instead of ACA, CAU, instead of UAC, ACC, instead of CGA, GAU. And the proteins you're going to get, or the amino acids you're going to get in that protein, are completely different. Now you have histidine, threonine, aspartate, and histidine, completely different from the original sequence. And the same thing would happen if you had shifted in the other direction. So you had started with something AU instead of AUG. You would get a same result where every single amino acid is wrong in that protein. So as you can imagine, that protein is not going to function. After initiation is complete, you have translation elongation, where you're basically building that polypeptide. So this is basically the same for all organisms. You don't have big differences for archaea or for eukaryotes. Every ribosome has 3 sites inside of it where a tRNA would be able to come in and base pair with a codon on the mRNA. They're called the A, P, and E sites. A for acceptor, P for peptide, and E for exit. So you start out with a tRNA that's in the peptide site, the P site. That would be your initiator tRNA that is attached to a methionine. Then at the A site, okay, here's my pointer. Right, so here's your initiator tRNA in the P site. At the A site is where new tRNAs would come in. Then once you've made a peptide bond between the amino acid on this new tRNA and the one that you already had in there, then the ribosome is going to shift. It's going to kind of walk down the mRNA, and everything is going to move. And your old tRNA that was in the P site will end up in the E site, and it just leaves. Any tRNA that enters the E site leaves, exit site. And you basically will go through this series of steps over and over and over again until you get to the end of the mRNA transcript. Well, I should say until you get to a stop codon. And this process also costs GTP at several points. So first, you have your new tRNA come into the A site. It's going to base pair with that codon. So in actuality, what's going to go on is you have multiple tRNAs that are all kind of floating in. They try to base pair. If the anticodon isn't right, they don't base pair. They just float back out. And pretty soon you get the right one floating in that actually does base pair successfully with this codon. And then you can move to the next step. So once this tRNA, a new tRNA, has entered the A site and successfully base paired with the codon there, then you spin some GTP, and the ribosome is going to catalyze peptide bond formation between these amino acids that are on the tRNAs in the P site and the A site. And it's actually rRNA that does this. So the protein parts of the ribosome do not do this catalysis. The rRNA that's in the ribosome is responsible for catalyzing the formation of this peptide bond. So rRNA is really doing most of the work in the ribosome, not even the protein. After this bond has formed, you're going to have the amino acid that was on the P site tRNA detaching from that tRNA. It's now attached by a peptide bond to the amino acid that's on the A site tRNA. And that whole thing is now just attached to that A site tRNA. So all of your amino acids in this chain that you're building, however many you have so far, will end up attached to the A site tRNA. Now the ribosome is going to walk on the mRNA, and that costs GTP as well. We technically call that translocation. So we say that the ribosome translocates on the mRNA. It's going to move. It's going to move down towards the end of the mRNA by 3 bases. It always has to make sure it moves by 3 bases each time, so that you're changing which codons are in each site, so that the A site codon ends up in the P site ends up in the E site, and so on. The tRNAs are going to remain attached to whichever codon they are base paired with, which means that they shift as well in terms of which site they're on in the ribosome. So you start out with this with this P site tRNA. After the ribosome translocates or moves, it's going to end up in the E site, and then it just leaves. The A site tRNA is going to end up in the P site. And now you're basically ready for the next round. For the next round of this to occur, you're going to have another tRNA come into the A site. You're going to add a peptide bond between the amino acid on that new guy and your existing P site polypeptide chain. Then the ribosome is going to translocate. Everything is going to shift its position. You'll have your old tRNA leaving. And you end up with a bigger and bigger and bigger polypeptide chain with every time that this process repeats. So that's the basics of that process. In addition to that, you also have a number of other proteins that have to come in and associate with the ribosome for it to work. Those are called elongation factors. We're not going to worry about them, but you know, they exist. So elongation continues until you get to a stop codon. The first stop codon that the ribosome reaches, that's going to be where it stops. When you have the stop codon enter the A site of the ribosome, you don't have a tRNA that can come in there. There is no tRNA that can base pair with the stop codon. Instead, you have another protein that's called the release factor that comes in. The shape of the release factor is very similar to the tRNA shape. So it's able to enter that A site similar to how a tRNA would, and it's able to bind to the stop codon. Once the release factor protein has bound to the stop codon, basically the whole thing disassembles. So your tRNAs that were in the E site or P site, they're going to leave. The polypeptide chain is going to be detached from the P site tRNA, and it's going to leave. And then your ribosome large subunit is going to leave, and the small subunit will also leave. So the whole thing just comes apart once you have release factor attached to the stop codon. And then just finally keep in mind that this is still a polypeptide chain. You're not done building the protein yet in a way, because it's not technically considered a protein until it folds up correctly and becomes functional. If you're a eukaryote, that folding would occur, of course, in the rough endoplasmic reticulum. If you're a prokaryote, it just happens right there where you happen to be, in the cytosol. So since eukaryotes have a nucleus, this makes the process of building a protein a bit more complicated. For prokaryotes, you're going to have transcription happening in the nucleoid, where the DNA is stored at, and you can actually have translation happening right there at the same time as well, since everything is just kind of in the mix together in a prokaryote. In the eukaryote, you have your DNA in the nucleus. That means you do transcription in the nucleus, but then you can't do translation there. First, you have to export the mRNA out of the nucleus, which could also require some ATP. And then once the mRNA is in the cytosol, or the cytoplasm of the cell, then you can have a ribosome attached to it and translate it. So this means that eukaryotes are going to translate or synthesize proteins a lot more slowly than prokaryotes could do. In eukaryotes, you make an mRNA transcript, then you export it, and then you translate it, and then you have a protein once it folds in the rough endoplasmic reticulum. In prokaryotes, you can actually have all of that happening at the exact same time. So here, this is like an electron micrograph image of transcription and translation in a prokaryote happening at the same time. This little line that you can see, that you can barely see, that is your DNA actually. And in this diagram version, here it is, here's your DNA. And you're going to have little RNA polymerases moving along it, transcribing the gene. This would be a particular gene. Right, so you're going to have maybe the promoter at the front here. So RNA polymerase is going to come in and attach and start transcribing. It's going to move along the gene as it transcribes, and the mRNA coming out of it will get longer and longer and longer as it moves and transcribes. Before it's done, though, you could actually have another RNA polymerase come in behind it and start transcribing again. So that's what's happening here. Every time you see one of these little chunky chains, kind of, that is mRNA that's coming out of an RNA polymerase. And you can see it much better in the diagram, of course. So here's different RNA polymerases that are all in the process of transcribing this same gene. And in red, you have the little mRNA that's coming out of them. So you can see the one that's closest to the start of the gene has the smallest mRNA. The one that's closest to the end has the longest mRNA. And they're all simultaneously transcribing the same gene into mRNA. At the exact same time, you have these ribosomes that come in and attach to the mRNA. As soon as you have a ribosome binding signal transcribed into the mRNA, you can already have ribosomes attaching there. So all these little blobs, these dark kind of blobs or balls in this electron micrograph, those are ribosomes. They're huge. So every one of these mRNAs is absolutely studded with ribosomes, and every ribosome is going to be making the polypeptide chain, right? So you have this ribosome starting at the bottom where you have, well, which should actually be the start, right? Where you have your start codon and your RBS. Then it's going to start translocating up the mRNA, heading towards the polymerase. But the polymerase, of course, is at the same time walking down the gene, and the mRNA is getting longer as it goes. And you have the polypeptide chain that's coming out of each of these ribosomes. And it gets longer the closer the ribosome has walked up to the polymerase. So this prokaryote is able to make like a bunch of copies of protein in one, like kind of simultaneously, basically, right? Every one of these chains is going to be a protein, and it's making them all at the same time. So this prokaryote decided to turn this gene on and express it, and already it's making many copies of that protein. In the eukaryote, it's way slower. You can have multiple polymerases making multiple transcripts of the same gene at the same time. That can happen. But then the mRNA has to be completely finished and exported out of the nucleus before a ribosome can start translating it. So the whole process is going to take a lot longer in eukaryotes, which does give prokaryotes an advantage because it lets them respond to their changing environment much more quickly than eukaryotes can do, at least in terms of starting to synthesize new proteins. Okay, so the final little bit that we'll talk about is protein processing and targeting. Protein processing mostly meaning like how you're folding that protein up, and then targeting meaning how you're getting it to the place in the cell where it needs to go. So the main part of processing is folding. There's also other things that occur in protein processing, but we'll just worry about the folding really. Folding for proteins can happen on its own. If you have just this polypeptide chain, it can fold up by itself. And a lot of the time it'll make the right secondary structures, but it might not make the right tertiary structure because that's a very complicated structure. For any given polypeptide chain, there's actually most likely going to be multiple ways that it could fold up into a tertiary structure, but only one of them will function. And this process of spontaneous folding can take a really long time, and you need it to happen quickly so that your protein becomes functional and is a true protein, a true functional protein, like, you know, as fast as possible, basically. So cells are going to use these special molecules called chaperones to make sure that proteins fold correctly. Actually, the chaperones aren't necessarily folding the proteins themselves, but they are almost like catalyzing the process. They're making the process happen much more quickly. It's not technically catalysis because it's not technically a chemical reaction. But also, in addition to making it go faster, they can prevent certain wrong foldings that are like maybe more likely to occur. They can prevent those wrong foldings from occurring, which overall just makes sure that your proteins are folding the right way and they're folding pretty quickly. You actually have some chaperones that are able to refold the denatured protein. So a lot of the time, once a protein is denatured, that's kind of it. Like there's not a way to fold it back up right. But in some cases, you have a chaperone that would be able to refold some of those denatured proteins so they can regain their functionality. An example of that type of chaperone would be the heat shock proteins, abbreviated HSP, which you will definitely hear a lot about if you continue in like molecular biology. Heat shock proteins are proteins that cells make in response to heat. So if a cell is exposed to temperatures that's higher than what it's like normally used to, it'll start making heat shock proteins. So it'll turn on gene expression for those genes that code for the heat shock proteins. Those proteins are chaperones. They're going to find proteins that are being denatured by the high heat, and they're going to refold them correctly. So basically meaning that the cell is not so vulnerable to high temperatures. The main reason why high temperatures, or one of the main reasons why high temperatures is bad for cells, is because it causes proteins to denature. And when proteins are denatured, they don't function. So these heat shock proteins are basically reversing that process and retaining the functionality of those proteins for the cell. Chaperones can also do other things in addition to folding proteins, like they could help different subunits assemble to make a protein that has quaternary structure. Sometimes they can also make sure that cofactors end up, joining with their enzymes in the right way. They can do a lot of things like that, helping different things in the cell like associate correctly or have the correct shape. And so in this diagram, we have protein synthesis, so that's transcription and translation, and at the end you get this unfolded polypeptide chain. If you don't do anything with that, it might slowly fold into the correct shape, or it might fold into the wrong shape and end up as an aggregate, which is just like a bunch of proteins that are misfolded or denatured all together in like a clump. And that can get those clumps can get kind of dense, and then it's hard for the cell to like clean them up once they're too big and too dense. It can actually be really deadly. So for instance, like mad cow disease and Alzheimer's, those are diseases that involve protein aggregates that the cell cannot get rid of and eventually get big enough to start killing the cell. So you want to prevent that from happening. And the cell is going to do that using chaperones. So what should happen is this unfolded polypeptide chain gets picked up by a chaperone that folds it quickly into the correct shape. And then you have your folded protein that does things. And a final note that in eukaryotes, this would be happening in, as I had mentioned actually, in the rough endoplasmic reticulum. Right, so the rough endoplasmic reticulum is studded with ribosomes. So those ribosomes are going to be making the polypeptide chains. Then those chains get kind of squished into the rough endoplasmic reticulum as they're built, and then the proteins would fold there. If we're talking about a prokaryote, then of course that doesn't happen in the rough endoplasmic reticulum. It just happens in the cytosol or in the cytoplasm. Finally, the last part of processing your protein is targeting it to the correct location. You got to make sure it goes where it needs to go. For prokaryotes, that's usually not too hard because there's only one place where you might send something, which is outside of the cell. Otherwise, most proteins are just kind of floating around in the cytosol. If you're a gram-negative bacteria, not only would you need to get that protein, if it's a protein that needs to go outside the cell, not only would you need to get it outside of your cell membrane, you would need to get it out of the outer membrane as well, in some cases. Some proteins would just need to go to the periplasm, others would need to go to the actual outside, which is more complicated, of course, when you have to put it through two membranes instead of just one. You also actually have some prokaryotes that can directly secrete a protein into the cytosol of another cell, which usually is not a nice thing that they're doing. Usually that would be some kind of cell that they're trying to like kill or invade, and they're secreting proteins directly into it, like to help them get inside of it or to kill it. But that could also not be a bad thing. Sometimes prokaryotes will do that to cells that they're living in like a happy symbiotic relationship with, and so they'll be putting proteins that this other cell finds useful into the other cell. In eukaryotes, it's more complicated because not only would you need to get some proteins outside of the cell, but you also have all these different organelles, and each organelle has a different set of proteins that it needs. So you have to make sure those proteins are going to the right organelle. In any case, wherever you're sending your protein, you're always going to target it to the right destination using a tag. And that tag is, there can be different tags actually, but a lot of the time it's an amino acid sequence that is somewhere in that protein, a special amino acid sequence. For prokaryotes, you would have what's called the signal sequence, which is just a series of amino acids at the front end of the protein that tell basically the cellular machinery to take that protein and put it outside of the cell. So you would have a whole system of proteins that are designed to carry other proteins across the cell membrane and or the cell wall. And the signal sequence allows a protein to kind of hitch up with those carrier proteins and be carried out of the cell. Another thing that the signal sequence can do is kind of prevent the protein from folding all the way. If it's going to be like kind of bulky and it needs to be outside of the cell, it's hard to get something big and bulky outside of the cell. It's easier to get it out if you only have it partly folded so it's more narrow. Then you can send it through your little tube that goes outside and then it finishes folding on the other side. In eukaryotes, a lot of the time you'll have amino acid sequences that are targeting the protein to its destination, but you can also have like different sugars that are added to the protein that send it to its destination. So in general, you would have proteins folding in the rough endoplasmic reticulum, then going to the Golgi apparatus. And the Golgi apparatus is going to add different sugars to them or make different other modifications. And those are all ultimately going to target that protein. They are going to serve as a tag that targets a protein to its correct destination. So you're going to have some carrier proteins that find that tag, and their job is to find something with a specific tag and take it to a specific organelle, and then they do that. And a final thing to note here is that anytime that you're moving proteins around or secreting them outside of the cell, that costs energy. So cells can use ATP as the energy source for that, but prokaryotes will also a lot of the time use the proton motive force as the energy source as well. So here you have different secretion systems for prokaryotes. Here's one that's just taking proteins outside of the cell membrane to the periplasm. Both of these are gram-negative examples. But here you have some other secretion systems that are actually sending that protein all the way to the outside of the cell wall. And on this side, you even have some that are sending that protein across the cell membrane of a host cell into a host cell. This one is sending a protein. This one's actually sending DNA into the host cell. And this one's like a little needle that's injecting the host cell with a protein. It gets kind of crazy. And then down here on the bottom, you have just a diagram showing that signal sequence. So the little red end of this protein is a special amino acid sequence. That is the signal sequence. And so any protein that has this is going to have these secretion carrier proteins that come in and attach to it and like excrete it through one of these secretion systems, and sometimes in an unfolded state.